Bismillahir Rahmanir Rahim. Welcome to Derma Immunology. I am Naglaa Elmongy, Professor of Dermatology, Cairo University, and our video today will be about neuroimmune interactions in the skin. Connections between the nervous and immune systems were known long time ago. However, recent research indicates that this communication and connection takes place more often than previously understood. The majority of interactions between the nervous and immune systems happen at specific anatomical sites where neurons and immune cells are found in close proximity. Neuroimmune communication coordinates functions like immune defense, tissue healing, metabolism, and thermogenesis. Understanding of the nervous and immune systems interplay traces back to inflammation's key indicators: swelling, redness, heat, and pain. Neurons can induce all these signs through neurogenic inflammation, and pain, the distressing sensation, frequently associates with immune reactions in inflamed tissue. Divisions of the peripheral nervous system, such as sensory, sympathetic, vagal, and enteric neurons, communicate with immune cells, affecting their functions. Central nervous system and the neuroendocrine aspects won't be covered here in this video. Instead, we will focus on the two-way interaction between the peripheral nervous system and the immune system, affecting homeostasis, inflammation, and illness. My reference in this video is this review article that was published in Annual Review in Neuroscience in July 2022, and its title is Neuroimmune Interactions in Peripheral Organs. Of course, I cannot discuss all the details that came in this paper, so if you need any more details, you can go back to this paper and read it, which is very informative. The molecular communication between neurons and immune cells affects neurological functions, barrier stability, and host defense, while also contributing to conditions like cancer and neurodegeneration. Both the peripheral nervous system and immune system are highly complex, characterized by cell types whose gene expressions depend on cell condition, tissue location, and interactions with nearby cells. At barrier locations like the skin, gut, and lungs, immune cells frequently reside near peripheral nervous system nerve fibers and have receptors for neuropeptides and neurotransmitters, making them receptive to neuromodulation. Similar to immune cells, neuronal cells also show significant diversity, undergoing molecular alterations following tissue injury and inflammation. Also, neurons express immune-related receptors like pattern recognition receptors and cytokine receptors, encouraging reciprocal responses and shared effects through these common receptors and mediators. We can see here in this diagram the immune cell and the peripheral neuron. So we have here in peripheral neuron, we have receptors for microbial products and pore-forming toxins, receptors for microbes, and pathogen-associated or damage-associated molecular patterns, and receptors for lipid mediators and cytokines and chemokines and growth factors secreted from the immune cells. Also, on the other side, we have immune cell have receptors for neuropeptides and neurotransmitters, including adrenergic and cholinergic receptors. Somatosensory neurons within the dorsal root ganglia and trigeminal ganglia innervate barrier locations like the skin and gut, transmitting sensations like touch, pain, and itch. Vagal sensory neurons, on the other hand, innervate internal organs. In response to inflammation, sensory neurons detect immune mediators and release neuropeptides such as calcitonin gene-related peptide and substance B and vasoactive intestinal peptide. These neuropeptides affect immune cell activity and affect host defense in the skin, lungs, and gut. Inflammation precisely modulates the sensory nervous system. Dorsal root ganglia and trigeminal ganglia neurons react to immune factors, triggering feelings of pain and itch. And while pain serves an acute protective role by promoting avoidance and aiding in wound healing, it turns into pathological state when it becomes chronic. In times of injury and infection, immune cells release a mix of substances like cytokines, histamine, lipids such as prostaglandins and leukotrienes, and growth factors like nerve growth factor. Nociceptive sensory neurons responsible for pain perception have the ability to detect most of these types of immune molecules. Several of these factors can induce neuronal sensitization and trigger action potentials, leading to pain. Subsequently, a local axonal reflex will give rise to the release of neuropeptides from peripheral terminals, contributing to the onset of neurogenic inflammation. Examples of pain-associated cytokines include interleukin 1 and interleukin 17 generated in chronic inflammatory conditions. Nociceptive neurons detect interleukin 1 beta via interleukin 1 receptor signaling. Interleukin 1 beta enhances neuronal excitability by reducing the action potential firing threshold. Interleukin 17, which is vital for defending against extracellular pathogens and linked to multiple sclerosis, triggers pain following injury. Vagal sensory neurons transmit sensory signals from the gut, lungs, and visceral organs to the brainstem, initiating sensory autonomic reflexes. Similar to dorsal root ganglia neurons, vagal neurons also have receptors for immune molecules. Again, here, the same diagram, we can see the immune cell and the peripheral neuron, where we have receptors in the peripheral neuron for microbial products and pore-forming toxins, microbes, and for receptors for pathogen or damage-associated molecular pattern and lipid mediators, and cytokines or chemokines and growth factors. And on the other side, in immune cells, we have receptors for different neuropeptides and neurotransmitters. And these receptors on peripheral neurons can transmit sensation of pain and also can result in axonal reflex arc, resulting in sensory autonomic reflex or synaptic release of neurotransmitters and neuropeptides. As regard the role of dorsal root ganglia sensory neurons in the skin's immune response, the skin is in continuous contact with environment, and it acts as a pathogen blocking barrier and a sensory interface connected to dorsal root ganglia sensory neurons. These neurons communicate with immune cells to control infection outcomes, allergic reactions, and wound healing. During infection, dorsal root ganglia sensory neurons connected to the skin interact with microbes and immune cells, controlling local immune responses. Microbes can directly impact nociceptive neurons through pathogen receptors, triggering pain. For example, nociceptive neurons have receptors for microbial molecules like formulated peptides and toll-like receptors. In response to bacterial signals, sodium channel Nav 1.8 positive nociceptive receptors release calcitonin gene-related peptides. This molecule then suppresses the production of tumor necrosis factor alpha in macrophages, leading to reduced recruitment of neutrophils and monocytes to the infection site, suggesting that neurons play a role in negatively regulating adaptive immunity. Sensory neurons also play a role in antifungal defense. Sensory neurons expressing what's called TRPV1, which is an abbreviation of transient receptor potential cation channel subfamily V member 1, are triggered by cell wall components from the fungal pathogen Candida albicans, carrying pain signals through the dectin-1 beta-glucan receptor. Sensory neurons release calcitonin gene-related peptide acting on dermal dendritic cells to stimulate interleukin 23 production, which in turn will promote the growth of dermal gamma delta T cells, resulting in interleukin 17 secretion and regulation of fungal load from Candida albicans. It was found that optical activation of channel rhodopsin expressing TRPV1 positive neurons triggers skin inflammation via type 17 inflammatory reactions involving the activation of gamma delta and CD4 positive T cells. Also, it will result in neutrophil infiltration and elevated levels of interleukin 6 and 23 and tumor necrosis factor alpha. This inflammation subsequently will lead to the management of Candida albicans and Staph aureus infection in the stimulated area and surrounding tissue. From the opposite side, also microbes can divert TRPV1 positive neurons to induce immune suppression and facilitate pathogen survival. And as an example is Streptococcus pyogenes, which is responsible for necrotizing fasciitis. This bacteria generates streptolysin S toxin that triggers TRPV1 positive dorsal root ganglion neurons, causing pain and inhibiting neutrophil recruitment. As regards skin allergic inflammation, sensory neuron and mast cells interactions are vital in allergic inflammation. Mast cells form close clusters with sensory neurons in the skin, containing densely packed vesicles full of molecules like histamine and heparin that result in inflammation, swelling, and edema. Activation of TRPV1 positive sensory neurons rapidly triggers mast cell degranulation through the neuropeptide substance P binding to the mast cell receptor, mast cell related G protein coupled receptor member X2, which is abbreviated by MRGPR-X2. Also, dorsal root ganglia sensory neurons aid in wound healing. Tissue-resident macrophages contribute to either inflammation or tissue repair based on environmental cytokines and signals. C-fiber sensory neurons with what's called G-alpha-I interacting protein or GINIP expression have protective effects in a mouse ultraviolet burn model. And deleting this GINIP positive fibers resulted in heightened fibrosis, elevated inflammatory macrophage counts, and reduced dermal anti-inflammatory macrophages. The neuropeptide TAFA4 produced by specific GINIP positive neurons controlled interleukin 10 production by dermal macrophages. So, sensory neurons in collaboration with immune cells can promote either protective or harmful dermal reactions based on stimuli. Now I'm going to talk about neuroimmune interactions in adipose tissue. Actually, neuronal signals extensively regulate adipocyte function, and research indicates that fat metabolism is affected by neuroimmune cell partnerships, including tissue-resident macrophages. A newly identified type of white adipose tissue macrophages is found alongside sympathetic nerve fibers. These macrophages are named sympathetic neuro-associated macrophages, which eliminate norepinephrine using the norepinephrine transporter SLC6A2 and the degrading enzyme monoamine oxidase A. As a result, these macrophages work to eliminate norepinephrine from the interstitium, consequently reducing the norepinephrine supply accessible to adipocytes. Typically, norepinephrine triggers adipocyte lipolysis and promotes fatty acid oxidation, initiating the formation of beige adipocytes through a process known as browning. Removing norepinephrine transporter SLC6A2 in sympathetic neuro-associated macrophages lead to white fat browning and heightened thermogenesis resulting in weight loss among obese mice. Also, a recent study noted that reduced lipolysis in adipose tissue adipocytes due to decreased noradrenaline levels with aging. Age-related changes showed increased expression of enzymes that break down catecholamines, potentially accounting for decreased lipolysis in older individuals. Similarly, tissue-resident macrophages were identified in brown adipose tissue, which is also sympathetic fiber innervated, and it was found that mutations uh, in the what's called MECP2 transcription factor within macrophages were linked to metabolic imbalances due to reduced sympathetic fat innervation. Brown adipose tissue macrophages consequently affect sympathetic innervation, indirectly regulating catecholamine availability. So collectively, these investigations highlight how tissue-resident macrophages maintain tissue equilibrium by transmitting neuronal signals. Lately, interactions between the nervous system mesenchymal stem cells and innate lymphoid cells type 2 have been revealed to jointly manage metabolism and obesity. As a conclusion, these neuroimmune hubs have served as central points that integrate diverse environmental signals to keep or restore tissue balance. These hubs present an interesting focus for innovative therapeutic approaches. Actually, the vagus nerve has been targeted through electrical stimulation. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell if you are interested in getting any new update, and thank you.